So yeah, I'm going to talk today about work that I've been doing. Really stretches back all the way to the beginning of my PhD and up to sort of work we're doing in my group now on the topic of polygenic adaptation, and then in particular on some challenges we've had in human genetics dealing with issues of population structure and some of the difficulties that creates in our situation where we can't do experiments, we can't do common gardens, and so we have to try to go out and measure phenotypes as they exist, out in the wild, so to speak, among the human population, and sort of how that problem or that peculiarity of humans creates some problems for us in trying to learn about polygenic adaptation. So the workhorse for us is this tool called a polygenic score, which I suspect most of you are probably familiar with at some level at this point. Polygenic scores are sort of an estimate of the so-called genetic component of an individual's phenotype under the assumptions of the sort of standard additive model of quantitative genetics. And so they have many potential uses. So perhaps the most well-known or most well-funded application of polygenic scores is in sort of clinical and medical applications, where you might hope that you can predict the disease risk of individuals from their genotypes, and thereby prioritize individuals for treatment or for medical screening or something like that. But they also have become a very powerful tool in basic biology research in allowing you to predict phenotypes of research individuals for whom you have not necessarily actually measured the phenotypes. And people have started to do very creative things with that sort of information. They've also shown up in the social sciences as sort of social science variables in predictors of social outcome phenotypes, which have genetic influences on them. I think this is a particularly interesting and also particularly fraught area in which people are trying to use polygenic scores, because it really sort of can violate a standard biologist's notion of what a phenotype even is. But then the scenario that I'm going to focus on, that I have focused on, that I'm going to focus on today, is in our study of evolution and adaptation, and how polygenic scores can enable us to learn about the history of adaptation for polygenic traits, but then some of the challenges that we've encountered in trying to do that. And then I'll end the talk sort of by talking about a path forward that we're trying to develop here. So I've sort of given a pre-outline. Here's the outline for the talk. I'll start by talking about sort of why polygenic scores can be useful for studying adaptation, particularly in humans. I'll give you an empirical example of struggles with this thing that we call stratification bias in polygenic adaptation tests, and I'll explain more about what that is as we go. Then I'll have a section of the talk that's sort of really a block of just pure theory, sort of examining how precisely and why this phenomenon of population stratification causes problems for polygenic adaptation tests. And then at the end, I'll present a little bit on sort of a new procedure that we've been developing that we think can sort of overcome the problems that we've encountered with stratification biases in these polygenic adaptation tests. So to set the stage, let's just talk about polygenic adaptation, and how polygenic scores are useful for understanding polygenic adaptation. So I'm showing you two slides here, or two sort of simulations here of allele frequencies evolving over time. And on the right, or sorry, on the left, there's a single trait or a single locus which contributes to a trait that's under positive selection. And then the remainder of the allele frequency trajectories are all neutral. On the right, there's 20 loci that all contribute to a single trait that's under positive selection, and another 20 loci that are all neutral. And so in the two cases, the strength of selection on the phenotype is the same. But on the left, I think that you can all probably pretty clearly see which trajectory belongs to the positively selected site, whereas on the right, you wouldn't have been able to pick out the signal of selection by looking at these allele frequency trajectories. And so this sort of is a fairly intuitive, I think, demonstration of the fact that when selection acts on sort of single gene traits, you get these large responses at individual loci, and that's responsible for our ability to detect signals of adaptation using FST outlier tests, selective sweep statistics, and things like that. But on the right, there's no individual site that stands out, and so our standard FST-like sort of methods or sweep-based methods aren't going to detect these signatures. However, if we take all of the sites that contribute to the trait under selection and add them up, add their contributions to the trait up, so weighting by the effect that they have on the trait, this is our polygenic score here. This would be like the average polygenic score for a population. Then we see this clear pattern of the polygenic score increasing in a way that is much faster than, or the change in the polygenic score from generation 0 to generation 400 is much larger than the change that you see if you take random sets of 20 sites and add them up to compute random polygenic scores. So the lesson here is that if we know which sites to look at, then we can compute these polygenic scores and develop tests which can tell us about which traits show evidence of adaptation in say human populations using population genetic data sets. So of course the catch is that we need to know which sites are associated with a particular trait, right? So this is a sort of, I like to think of this as a sort of forward genetics approach to studying adaptation, and that the first thing that we do is we use genome-wide association studies, where you plot phenotype against genotype, and you look for sites where there's a non-zero association between genotype and phenotype, and estimate an effect size for an individual site using just standard linear regression sort of techniques. Then you compute a polygenic score in a new test panel, and then you test whether that distribution of polygenic scores is consistent with neutral evolution, relying on the fact that polygenic scores are just sums of allele frequencies. We understand how allele frequencies evolve under neutrality, and so we can leverage this into a test for selection. And so what I'm showing you here is a figure using GWAS data from a GWAS for height that was done by something called the GIANT Consortium. This was a huge meta-analysis consortium in human genetics comprising like 80 different labs around the world, mostly Europe and the US. And we've computed polygenic scores for a bunch of different populations across Europe. The x-axis, which I failed to label here, is latitude. And you can see that the polygenic score for height increases as you go to the north in Europe. And if you perform a test for polygenic adaptation, you can reject neutrality as an explanation for this pattern. Now, this plot comes from a paper of mine published in 2019. This signal is actually much older than this. Dates back to 2012 in this paper by Michael Turchin and Charleston Chang, who, using an earlier iteration of this GWAS, showed that height increase in alleles are systematically at higher frequencies in Northern Europe than in Southern Europe, according to this particular height GWAS. And that was sort of the first demonstration of this signal of supposed polygenic adaptation for height. Now, a bunch of papers published on this signal over the span of the last decade, starting with this Turchin and Chang paper in 2012, and going up to this paper that I was about to publish in 2017 before things sort of went sideways. And so I'm going to show you one figure from this paper in 2017, just to show you kind of the potential promise of polygenic scores for helping us learn about the history of adaptation in human traits. And then I'll shift to telling you about some of the problems that we encountered. So here is an analysis done by Xinjun Zhang, who was a fellow PhD student at UC Davis when I was there, showing polygenic scores for height using these same effect sizes computed from this giant consortium meta-analysis. And these are computed in ancient individuals, which are the gray dots you're seeing here, as well as in present-day 1,000 genomes individuals, which is what you're seeing stacked up on the right here. And so what we noticed in these plots, and we confirmed this via sort of formal statistical tests, is that there are these really old ancient individuals here that have relatively low polygenic scores, particularly in comparison to the European individuals to whom they contribute ancestry. And then there are these more recent ancient individuals, most of these individuals coming from various places across Europe, who have dramatically increased their polygenic score for height to levels sort of in line with what we saw in this data set in modern Europeans. And so this horribly messy, very non-scientific figure is my sort of attempt at reconstructing the network of connections among all of these ancient populations that we were looking at the time. And the important thing to note is that highlighted in red are all of these samples that sort of correspond to these ancient populations with elevated polygenic scores. And blue are these ones that were in the past and were much lower. And what you can see is that there are sort of some of these samples highlighted in red that fall on either side of this very deep split in this tree, which suggested to us that there had been selection for increased height along two separate sort of branches in the Eurasian sort of admixture graph or population tree of human populations, suggesting two different episodes of selection on height. So this was super exciting, and we were very much looking forward to publishing this. And then just as we were in review, the UK Biobank resource was published. And so the UK Biobank is this fantastic sort of GWAS resource, 500,000 individuals from the UK, genotyped across the genome and phenotyped on many, many different traits, including height. And so we said, well, let's replicate the signal that we've been seeing with this older meta-analysis GWAS using this UK Biobank resource. And so here is this figure that I've already shown you, demonstrating how polygenic scores using this older GWAS data set increased as we move to higher latitudes in Europe. We saw a similar, in fact, even stronger signal of polygenic scores for height decreasing sharply as you move from west to east across Eurasia, which we interpreted as well as another signal of polygenic adaptation we thought sort of the same events, the same ancestral selection events. However, when we compute the polygenic scores using the UK Biobank resource, we see basically no signal. So each of these columns here corresponds to sort of a slightly different way of doing the GWAS in the UK Biobank, just swapping out which exact individuals you include, which precise methods you use to try to control for population structure. This column on the right here is a method that uses only comparisons within siblings that should be totally immune to population structure. And really, no matter which way we did this test, these really strong signals that we had seen before pretty much completely dry up when we use this UK Biobank resource to compute our effect sizes here for our polygenic scores. So this was discouraging and definitely worrying. And so we dug into it a little bit, and what we found is that we think the effect sizes generally do replicate across these two data sets. In this figure here, I'm showing you effect sizes from this earlier GWAS, this giant consortium GWAS, plotted against effect sizes from the UK Biobank, and you see that on balance, when you have a positive effect in the earlier GWAS, you also have a positive effect in the UK Biobank GWAS. But when we plot the difference in allele frequency between a Northern European population, like individuals from Great Britain, minus the allele frequency from individuals in a more southern population, like those in Tuscany, Italy, and we plot that against the difference in effect size between these two studies, we find that these two variables are correlated. And what that suggested to us is that these effect sizes in this giant consortium GWAS here perhaps are biased, and that they're biased by population structure. That the further north you go in Europe, or sorry, let me say this right. If you look at sites that have larger frequency differences between north and south, that their effect sizes seem to be more biased towards being positive in this giant consortium GWAS relative to the UK Biobank GWAS. Another demonstration of this came from another paper published at the same time as ours by Michal Sohail and Robert Meyer at the Broad Institute, then at the Broad Institute. And so on the left here, you're seeing minor allele frequency. The axes are the minor allele frequency in Great Britain and the minor allele frequency in Tuscany. And the coloring is showing the average effect size estimate in a given joint minor allele frequency bin. And so what you're seeing is that all of the sites that fall above the diagonal here, that are at higher frequency in the north than the south, have systematically more positive effect size estimates in the giant consortium, whereas the reverse is true for sites that are at higher frequency in the south and the north, and that this isn't true in the UK Biobank. And so this is a form of what we call stratification bias, that what we're almost certain happened in this giant consortium GWAS is that there are associations between environment and an environment that has an effect on the trait. This T here and this blue circle stands for the trait. So there's associations between the environment and the trait that create differences between groups. And there's also associations between sort of ancestry and the trait because individuals are distributed spatially across the landscape. And so you get these sort of correlations between ancestry and these environmental effects on the trait, and that leads to biases at individual SNPs, which may or may not affect the trait in terms of their the biases in terms of their effect size estimates. I'll explain this more as we go into specific examples or specific theoretical examples. And so the problem then is that GWAS that don't sort of fully control for this correlation result in biased effect sizes that are systematically biased with respect to ancestry. And because our polygenic adaptation tests rely on testing for differences between groups that have diverged in ancestry, this sort of can create these biases in our polygenic adaptation tests. So I'm in this talk focusing specifically on problems with adaptation tests, but they're of course a problem, beyond just selection tests, in the sense that if you wanted to use polygenic scores to make predictions about phenotypes of individuals and then use them for something, say for medical purposes, you had better be sure that those predictions are accurate, right? That they're not biased with respect to ancestry, because then whatever downstream whatever you're doing downstream of the computation of those polygenic scores will sort of recapitulate those biases. And particularly if ancestry is in the mix, that can be a real problem if you think about sort of social stratification and correlations with ancestry that exist in many societies. I think I'll skip this slide. Okay, so that sort of brings us to where we were about 2 years ago or so. And then there's this paper published by Charleston Chang and Minhui Chen. I think this was in 2020, where they wanted to go back and look at, again, some of these signals of polygenic adaptation for height in Europe. And in particular, they wanted to look at Sardinia, because individuals on native Sardinians are in fact significantly shorter than mainland Europeans. This was one of the first sort of signals of polygenic adaptation that was reported, was a contrast between Sardinians and mainlanders. And so there was some thought that perhaps this signal was real, even though these sort of broad continent-wide signals had not been, or seemed to be the result of bias. And so what they did was something very clever, was that they used GWAS effect size estimates taken from the Biobank of Japan rather than from European populations. And so when they did that and they compare Sardinians, which is this dot right here, to mainlanders, they do find a significant difference between them, sort of rescuing some amount of this evidence for selection on height within the history of European ancestry populations. Now, why was this thing of choosing effect sizes from the Biobank of Japan a good idea? So what they're doing is that they're sort of hoping that that they have a sort of separation of population structure between structure in Europe and structure in Japan. So that if, suppose, there's hypothetically some environmental variable that increases height as you move from south to north in Japan, that could bias the effect sizes, but hopefully it doesn't bias the effect sizes in a way that is sort of correlated with patterns of allele frequency variation across the European continent. And so even if these effect sizes retain some amount of residual bias, the hope was that that these biases were sort of somehow orthogonal to the patterns of frequency variation that matter for these polygenic adaptation tests. So that's this sort of insightful thing that this one paper did. And now I'm going to spend the rest of my talk sort of building a theoretical justification for this procedure, and then sort of expanding on that theoretical justification to sort of expand it beyond this simple scenario where you have this total separation of the panel that you do the GWAS in and the panel that you do your polygenic adaptation test in.
Jeremy,
yes.
Can I just interrupt very quickly?
Yeah.
So, when you talk about the Sardinians here,
yeah.
So, there are two things, right? I mean, one is, the whole question of whether Sardinians are actually genetically or environmentally shorter.
Mm-hmm.
And then there's the selection one, right? So,
right.
So, I mean, right. So there's an extra layer of complication whether, so first, do you even believe that they're genetically shorter, or is that all bias? And the second one is, do you believe that selection has something to do with this? But I guess you're going to probably go into trying to disentangle those, maybe because it's like I have sufficiently hard problems. That's like, it's like I've hard enough problem understanding whether I believe in the betas for themselves, and then there's an extra layer of whether I believe in a population index model on top of that. Then,
right. Yeah, so that's a good question. Let me try to answer it this way. I think that the question of so there's a question of are Sardinians, say, genetically shorter
in some sense, which also needs defining. Yeah, but
yeah. So that's a difficult sort of causality question, right? About like, do we believe these polygenic scores sort of almost as a causal model for the phenotype in some sense?
Right.
And that's a very difficult question, I think. Whereas the selection,
but at least that one is sort of in principle kind of experimentally. At least in plants, you know how to answer that one. You can just move people.
Yeah, exactly, right. This, in some senses, the selection question is almost easier because we know that under neutrality, these effect size estimates, sort of however they are obtained, as long as they aren't obtained by using allele frequency information, the effect sizes should have nothing to do with the way that allele frequencies have evolved across populations.
Sure, but they are, right? Because whenever you do a GWAS, allele frequency is in the estimate, right? So I mean, if you get the estimate from a GWAS, then the allele frequency is in there. I mean, I'd buy it if you estimated the effect sizes in a dilated cross, right? And then, you know what I mean?
Right. Yeah. So I think what I'm going to try to do in the rest of the talk is to identify precisely how allele frequency enters the effect size estimate, and hope that can show us a way to sort of remove remove the component of allele frequency variation in the estimate that leads to any potential biases.
Sure.
And so that's sort of the hope then is that will let us sort of recover this logic that I was arguing for, that that the effect sizes, and particularly the sign of the effect size, should have nothing to do with sort of the direction of allele frequency change, if we assume that the allele frequencies are evolving neutrally. And then that sort of allows us hopefully to recover some sense of that if we see an association between an effect size and patterns of allele frequency change, that is going to tell us something about selection, and not just bias in the estimates. The problem of that causal question of is this population genetically shorter in some sense, I think is a harder question that I'm not going to try to fully grapple with today. Okay, so to get into this, let's sort of take a step back and let's think about where these effect size estimates come from. And to do that, we're going to rely on sort of a simple, perhaps overly simple model where we'll suppose that our phenotype is generated by some true model, sort of hewing to the standard, models of quantitative genetics, where we have the effect of some site on the trait, and we have some confounding environmental variable, and then we have sort of all of the stuff in the background that could be contributions of other sites and could be other environmental effects that are not sort of systematic across the population in some way, that are sort of random noise. And then our GWAS model is just going to not include this confounding environmental variable, right? Like we just have the effect of our site and our residual error. So when we estimate the effect, then using this model that doesn't include our confounder, the true effect estimate is going, or the effect estimate is going to include three components. One is this true effect, the true beta, and then it's going to have this stratification bias term in it. And then an additional term that just describes the noise and the effect size that has to do with things like the sample size. So we can take this effect size estimate and break it apart into these three components. And now suppose that we do a GWAS in a simple scenario where we have two populations, A and B, that are diverged by some amount. And suppose that there's a difference in average phenotype between them, which I'll write delta AB. Now, assuming that FST between A and B is relatively small, we can write this stratification bias term for our effect size estimate as the product of a few different terms. One is this difference in phenotype between the two of them. And then it's going to be that times the difference in allele frequency between populations A and B at this particular site. And so this product here is just normalized by a numerical constant of 4, and then this sort of heterozygosity in the combined sample of individuals from the two populations. And so we can kind of ignore these terms on the bottom. They're not going to be super important for thinking about the issues here. The real problem comes from this. We have this effect on the phenotype, this environmental difference between the two populations, and then this allele frequency difference here. And so both of these things have a sign. And so if the signs are both positive, or if they're both negative, the bias and the effect will be positive, right? If the signs differ, the bias on the effect will be negative, right? So now suppose that we take two additional populations, C and D, and these are going to be our populations in which we do this polygenic adaptation test. We compute polygenic scores in C and D using our estimated effect sizes and our allele frequencies from these two populations, and we add them up to compute our polygenic scores. And then if we think about doing a polygenic adaptation test, we might compute something like QST for these these polygenic scores. So if anybody's not familiar, QST is just like a phenotypic analog of FST. It tells you about how much of the variation in a trait is among populations relative to the amount that is within populations, in just the same way that FST does this for an individual marker, right? And so you can see that in this sort of simplified example, or this simplest case, our evidence for polygenic adaptation comes down to a statement about the difference in average polygenic score between these two populations. So I'm just going to focus on this thing, and I'm just going to think about, well, what is the expectation of this difference? You know, assuming that we've got this bias term in here. And so we're going to have two components. One is the sort of true difference between the polygenic scores. That's the thing we're actually trying to measure. And we're actually trying to test whether this thing is non-zero, and specifically, is it sort of significantly non-zero? And then we've got an extra term that corresponds to this bias in the effect sizes. And in particular, it's this sum across loci of the product of these bias terms with the differences in allele frequencies between our two test populations, C and D. So if we take our expression for the bias here, and we insert it into this sum and sort of pull out in front the terms that aren't indexed by L, we get a term that looks perhaps somewhat familiar if you've paid any attention to the sort of demographic inference literature over the last 10 years, in that it looks very similar to an F4 statistic. So an F4 statistic first introduced, as far as I'm aware, by Patterson et al. In 2012, reviewed in this paper by Ben Peters. Fantastic paper that you should take a look at if you're not familiar with these statistics. But basically, these statistics measure sort of how much shared branch length is there in this A versus B comparison versus this C versus D comparison, right? And so it's this gold sort of branch that I highlighted here that this F4 statistic is measuring. And so now if you look at what we've got appearing in our expression for the difference in polygenic scores, assuming that this bias component is present, we've got something that looks an awful lot like that. It just has this sort of normalization by the heterozygosity in the denominator, but it's going to do sort of fundamentally the same thing, and that it's going to be some kind of measure of how much of this, how much of the population structure, how much of the allele frequency variation in these two different contrasts that we're interested in, the one that's in our GWAS panel and the one that's in our polygenic adaptation test, how much of that is shared between these two contrasts? How long is this sort of branch that I've highlighted in gold here? And so our measure of sort of the magnitude of the bias winds up being closely related to these standard statistics of population structure. And I'll just call this sort of like a modified F4 statistic and put a little tilde over it to acknowledge that. And so if we think about doing this GWAS and polygenic adaptation test with populations in this particular configuration, where we have A and B on opposite sides of this deep split and C and D also on opposite sides of this deep split, then this modified F4 statistic will be greater than 0, and we'll have non-zero bias in this test if we didn't control for structure in our GWAS. On the other hand, if we have a configuration that looks like this, where our GWAS individuals are coming from A and B, which are on this side of the split, and the polygenic adaptation test is being done among populations C and D, which are on this other side of this split, then this modified F4 statistic is 0 in this case, because if we go back and look at what it looks like, right, the differences in frequency, suppose that this was B and this was C, right, this was AB and this is CD. Suppose I had the tree that I showed you a few slides ago up here, then the differences in frequency between A and B would have nothing to do with the differences in frequency between C and D, under genetic drift, right? And so then this statistic winds up being 0. And so even though the effect size estimates are biased, they're biased in a way that is random with respect to the pattern of allele frequency variation that matters for the polygenic adaptation test that we want to do. So the biases essentially show up as noise in our polygenic adaptation test rather than signal. And so that justifies, that is precisely the justification that this paper was relying on, is that they are assuming or hoping that if there are environmental effects in this Biobank Japan population, that they sort of correspond to these like A-B-like comparisons in this particular tree, and that the Sardinia versus mainland comparisons that they're making sort of correspond to these C versus D comparisons, and that there is no overlap in population structure, so that even if these biases exist, they show up as noise in the adaptation test. Now, of course, the issue or an issue here is that they don't know that this is true necessarily, right? It's sort of reasonably well justified from our understanding of patterns of population structure and human, histories of human migration, but there's no formal test here that's showing that this is actually true. It's still a sort of, hope and worry kind of situation, right? So now, my question for the rest of this talk here is sort of, can we use the insights gained in these past few slides and in thinking about this particular test that they did to sort of generalize to cases where the GWAS and test panels might have some shared structure, or to and to sort of make this approach pioneered in this particular study airtight. And so I think the answer is yes, and I'll try to walk you through the approach here. So let's come back to our sort of simple model here, where we have some truth, we have some GWAS model, we have this problem of the effect size estimates being biased. And of course, the best thing you could do is just include the environmental variable. Well, the absolute best thing you could do is randomize, right? That you would do an experiment or a common garden, as Magnus says, take your plants, put them all in a common garden, and then you're good. Of course, we can't do that in humans. So the next best thing you could do is actually measure the relevant environmental variable and include it as a control. This would completely eliminate these biases, and then we'd be set. Of course, the problem is we don't know we don't know what the environmental variables are, or even if we do, we couldn't probably measure them in a way that would be sufficient. And so what people do instead is they include principal components, right? They go to the GWAS panel, they look at the top X principal components, where X is some number that you pick and hope is large enough, and you include these as covariates in the GWAS. And then if our environmental variable vector, eta here, can be represented as a linear combination of these principal components, then all of this bias will be eliminated, and you're okay. And of course, we have the same problem here, is that we don't know what the environmental variable vector is, and so we don't really have a way of knowing whether what we've done has worked. So we're in this situation where we want to include some kind of covariate here that's going to eliminate the biases, or at least eliminate the biases in the polygenic adaptation test that we want to do. And we need to figure out what this vector is. What is the covariate that we need to include, or that we something that we can actually figure out how to compute, that will immunize our polygenic adaptation test against these particular biases. So let's again think back to our simple scenario. Suppose we have two populations X and Y. We have this sort of simplest polygenic adaptation test you could do of computing QST and then asking, is QST larger than FST by some amount? And the key thing here is this numerator term, this difference in polygenic scores between the two. And so if I do what I did before, and I look at the expected difference in polygenic scores between the two, again, I have this, this true difference, the thing that we're trying to measure. And now what I want to do is I want to find a way to split the bias into two components. I want to split the bias into one component that behaves in the way that the biases did in this sort of non-overlapping structure scenario, or in this sort of Sardinia and Japan case that the Chen paper pursued, where there's a component of the bias that we can allow to remain in the effect sizes, as long as when we look on average across sites, that bias has sort of zero covariance with the differences in allele frequencies between the two populations that we're interested in. And then the remaining component of the bias, indicated by this sigma LC here, that we have to reduce to zero in the GWAS. Okay, so we can get this sum here to zero by having, we can allow these individual biases to be non-zero as long as the sum is zero. And then this component here, we need to reduce these individual terms here to be actually zero. Right, so that's what I just said. We need to identify some way to compute a covariate to include in the GWAS that reduces these ones to zero. So the way we'll do this is if we think about a matrix of genotypes in the GWAS sample, we have the matrix of genotypes in the test panel. And then if we think about our test as sort of specifying some test vector, so this can be, the test vector can be anything, right? It could be, in the simple examples I've been showing you, it's just been population ID. Are you in population X or population Y, or population C or D in the earlier example? So, Sardinia or mainland. But it could also be something like latitude, right? In the earlier examples that I showed you from the, the giant consortium tests that look to have been biased by population structure. So then we're going to compute sort of a projection of this test vector into the GWAS panel. So if the test vector is indicated by T, then I'll write this projection as just T GWAS. And then I include this projected test vector as our covariate. One way to think about what's going on here is that this is this collection of matrices, genotype matrices that I'm multiplying together here. You can sort of think about it as like a multivariate regression coefficient. So I'm giving the best prediction of this test vector in the GWAS panel based on the values that I have assigned to individuals in the test panel in order to do the test. And if I then include that as a covariate in our GWAS, that eliminates specifically this component of the bias that will create biases in our polygenic adaptation tests. It leaves behind the components of the bias that are uncorrelated with the patterns of frequency variation that we're looking at with this particular test vector. So you could also try to control for those additional biases as well, but it doesn't matter whether you do so successfully or not for the specific test that we're going to try to do. So just to sort of back up and review, the sort of standard procedure is that you do a GWAS with principal components of the GWAS panel as covariates. You compute polygenic scores in the new panel using the effect sizes that you obtained from that GWAS. And then you test for an association between polygenic scores and your test vector. And you ask, is this covariance between these two things different from zero according to some statistical test? And then you hope that the residual biases in the GWAS effect sizes have not biased your test. And so our procedure is a little bit different, right? The key difference here is that we have to identify before we do the GWAS, we have to identify specifically what test we're going to do and write down this test vector, which can represent population membership, geography or environment. It could represent time if you have ancient samples, anything you want, but you have to write it down before you do the GWAS. Then using both the GWAS panel genotypes and the test panel genotypes, we compute this projection of the test vector into the GWAS panel, conduct the GWAS using this vector as a covariate, and now it's the same as before. We compute our polygenic scores in the test panel using the GWAS effect estimates that we obtained in step 3 here. Now we can do our polygenic adaptation test, and importantly, now we can be confident that whatever biases remain in our GWAS effect size estimates won't bias the specific test that we're doing. So now I'm going to show you. So we've kind of got the theory and the logic of this worked out, and we're just to the point now of getting up and running in simulation. So I'll show you just in this very simple simulation scenario. We've done coalescent simulations in a 4-population tree where divergences are about FST of 0.1 between these for these lower divergences and about 0.2 if you go back to the root here. We assume that we simulated a phenotype with no genetic component. And we then create we simulate an environmental difference between populations A and B. We do a GWAS in panel AB, panel of individuals composed of A and B, and we do our polygenic adaptation test in C and D individuals. And so if we don't control for population structure, as the difference between the two populations increases, we get a higher and higher false positive rate in our polygenic adaptation test. But if we do this procedure of first computing the sort of version of the test vector in the GWAS panel and including that as a covariate, our false positive rate stays completely flat, no matter the magnitude of the environmental difference. Now, the thing that's the sort of honesty in advertising thing I should say here is that this is about the simplest possible scenario that we could set up, right? And that I could get this plot with standard methods as well, right? That if I just included the first principal component in A and B is going to separate individuals in A and B, and it's going to produce essentially the same plot. But in doing this, creating this plot here, we haven't done that, right? We've made no specific assumptions about which axes are important for the environmental variable. All that the method knows is what test we want to do. And because all of these polygenic adaptation tests can sort of always be framed in terms of these sort of simple linear models of just like testing for an association between the polygenic score and the vector that we're interested in, this linear algebra is general. And so we expect that this is going to hold for sort of arbitrarily complicated scenarios that we simulate, which is what we're working on now. Okay, and so with that, I just want to acknowledge Arbel and NASA, who contributed to the work on issues with stratification in the Giant Consortium, Xinjun Zhang, who did the ancient DNA analysis that unfortunately didn't pan out, Graham Coop and Jonathan Pritchard, who are senior collaborators in that work, the November and Steinrechen labs at U Chicago, who've heard many earlier iterations of this work, and then Jennifer Blanc is a PhD student in my lab who did sort of the second half of the talk. And so I'm happy to take any questions.